how the disease progresses or changes using various genetic and epigenetic modifying factors can shape the decisions that we use in the clinic. Dr. Spivak, thank you.
Richard. Well, first, let me express my gratitude to the patient committee for the opportunity to talk here today. It is a truism in academic medicine that doctors are always talking to other doctors. And when we talk to patients, and plural doctors, we generally talk to one patient at a time in our examining room. So it's very unique to have an opportunity to talk to and share thoughts with many patients at one time. I want to thank Richard Silver because he is the prime mover on moves behind this session. And finally, I have to thank John Crispino because he did all the heavy lifting, which will make it much easier for me in my talk because he's given you just an absolutely wonderful background. So let's start out with this cartoon, which is similar to what John showed you, and I show it because it's going to come up again, where we have the prime mover on move, the primitive stem cell, and actually we won't talk about it today, but we can identify that cell, we can isolate it as a single cell, and we think we can isolate the cell behind it. That's the Jack2 mutation occurs at the top at the primitive stem cell, which essentially makes itself and it makes daughter cells, which eventually lose the ability to renew themselves, and they become the cells that we see in the peripheral blood, the red cells, white cells, and platelets from megakaryocytes. Now, this is a second cartoon which talks about the evolution of clonal dominance. So we don't have just one stem cell in our bone marrow at any time. We have many, but only a few are making daughter cells and committed cells. And so what I show you here is one cell happens to transform. We don't understand why. It can be hereditary, it can be environmental, it can be a combination of both. And now you have a cell that has a better survival capacity than the normal stem cells. And what happens next, over time, you'll see all of a sudden we can't find the other stem cells. We have clonal dominance. Now, if you're up at the top of the slide where you have both normal and neoplastic stem cells, you have what we call a chimera. And generally, that keeps the neoplastic stem cells intact from taking over, but we don't understand why. In most cases, that doesn't happen. So with that background, we're back to these three disorders. And I can tell you now they're clonal stem cell disorders and that they exhibit clonal dominance. Eventually, the affected stem cell, the one with the Jack2 mutation, the MPL mutation, or mutations that we don't know about in 30 to 40% of the patients, leads to clonal dominance by varying degrees, and I will show you that. So blood production can be increased in these disorders, it can be decreased in these disorders, it can be outside the bone marrow in the spleen, and I'm going to show you phenotypic mimicry to explain why we did what we did in this study that I'll tell you about shortly. And then the big problem, we don't have specific diagnostic markers for these diseases, and there's a tendency, as Ron nicely pointed out, for genetic instability and disease transformation. So, before I go any further, I want to, this is a personal editorial, clear up some of the confusion in the field. And to start with, these diseases are not common, and they have diverse clinical manifestations, and they are not static. They evolve over time. Few physicians see many of these patients, and even fewer see them for long enough periods of time to actually recognize what's happening. Then, each of these is clinically different, but they can mimic the other disorders, and they can mimic benign disorders. So you have to really, there's big problems here, and again, there's no diagnostic test for any of them that's specific. Their natural history is not completely defined. And finally, unfortunately, the clinical perspectives about these disorders continue to be driven by unproved assumptions that are over half a century old. And I'm sorry to tell you that, but that's a fact. So here's something. What's in a name? 2002, the World Health Organization decided to get into the MPD business, much as McDonald's opens new franchises. This was their franchise. They are pathologists. They lumped the three myeloproliferative disorders with a bunch of other unrelated diseases. 2009, they decided we've got to do something new. They changed the name from MPD to MPN, which was not an advance. and they changed the name of idiopathic myelofibrosis to primary myelofibrosis. Well, it turns out there's no such thing as primary myelofibrosis. Myelofibrosis, as Dr. Crispino told you, is a secondary process. Stem cells are telling other cells what to do. It is a reversible process. It occurs in benign diseases. It does not, by one iota, affect the function of the stem cell. Primary myelofibrosis is all about a bad stem cell, and the fibrosis is just a manifestation of the bad stem cell. So let's go on. So why do I say this? Because here's 12 reasons why those 3 diseases do not belong in that classification. And we have a problem. Here's a 13th reason. These diseases mimic each other and they evolve into each other. So someone who thinks they may have so-called essential thrombocytosis, which really means thrombocytosis of unknown etiology, can morph in our series, 27% become polycythemia vera. 20% become primary myelofibrosis. Some patients with myelofibrosis develop polycythemia vera, and some patients with polycythemia vera will get fibrosis in the marrow. But essentially, all pathways lead to polycythemia vera, and pardon me for this, but I call this the Boy George syndrome. Looks like a girl, but it's a boy. So whenever someone comes with a myeloproliferative disorder, you have to think of phenotypic mimicry. So here's a patient, time on the bottom axis, with a high platelet count, and over a 6-year period, all of a sudden the hemoglobin went up. He developed polycythemia vera, not essential thrombocytosis. Here's a patient who had primary myelofibrosis for 17 years, and all of a sudden, even on hydroxyurea therapy, the hematocrit went up. This was an evolution to polycythemia vera. And here's another patient, I'm going to cut to the chase in the interest of time, who had been watched over a period of years as their platelet count went up, had a complete workup. The World Health Organization workup certified bone marrow, heterozygous for Jack2, normal iron. This woman has to have ET. No, she doesn't. Hematocrit is a little high. We did a red cell mass and plasma volume, and she had polycythemia vera, because in polycythemia vera, the plasma volume expands and it hides the number of red cells. So you have to watch out what's going on. Now, John Crispino went through this, and I don't want to spend time on this cartoon, but get back to the point of, in the myeloproliferative disorders, there is a Jack2 mutation, and this mutation allows cells to, multiply uninhibited because they don't need a signal. They already have a constant signal. So let's look at early cohort of our patients. And what you see here are the percentage of patients who had the Jack2 V617F mutation in PV, PMF, and ET. And you can see that in PV, 92% of the patients, well, actually, if you add the exon 12 mutation, you're going to come up with 98% or so of the patients had a Jack2 mutation. But the other 2 diseases didn't have the Jack2 mutation in all cases. So what's the story there? So here we're back to our classical stem cell model. And let's hit the button, and what do you see? Well, down at the committed progenitor cell level, you see the 4 receptors that require Jack2. And if all of those receptors are constitutively active, guess what you get? Polycythemia. Too many red cells, too many white cells, too many platelets. So essentially, what we can say is that polycythemia vera is the ultimate phenotypic consequence of the Jack2 V617F mutation, meaning this is the most that this mutation can do. It can make every cell line increase. Well, if it's the most that this mutation can do, then if you were a physician, you would immediately say, well, anytime I see someone with a Jack2 V617F mutation, what should I be thinking about? This disease. Unfortunately, that has not been the case. Now, how do we get one mutation to cause 3 different diseases? This has been a conundrum in the field, and the answer to the conundrum is really very simple. You can't. It doesn't, okay? Other things are going on. You've heard of the other mutations that are present. We don't understand this completely, but I will show you some of the things we do understand. So let's step back and talk about what are the genetics factors involved in disease manifestations. And disease manifestations, as Ron said, are what you see. You see me standing here, I've got a white shirt, striped tie, and a blue blazer. That's my phenotype because I'm in academic medicine, okay? Sorry. And so this will vary. Richard's more senior, he's wearing a suit. So, all right, so what you have is we have a DNA alteration. That's the disease-defining mutation. In this case, it's the Jack2 mutation. But then we have an unknown, patient DNA. As human beings, we're outbred, we're polygenic. That's why it's so hard to find, say, the cause of type 2 diabetes, because there's so many genes involved, or the disease schizophrenia. There are problems here. We don't know about patient DNA, and it's the interaction of the disease mutation and patient DNA that gives us what we call the phenotype. So you have genotype, patient genotype, disease phenotype. And in some patients, the disease is very penetrant. What do you get? Polycythemia vera. In other patients, it's not. What do you get? ET. So moving on from there, this is a hard slide to look at, but I've got the three diseases, ET, PV, and PMF. And what these are what we call the Jack2 mutational burden. It's not enough to know you're Jack2 positive or negative. We can figure out very simply how much, what's the clonal dominance or non-dominance. And if you look at the top box there, ET, everything's down in that lower left-hand quadrant because these patients don't get clonally dominant. They have an equal or more number of normal cells. Why does that happen? We do not know yet. And if you look at PV, you have a mixture, but most of the patients are clonally dominant. So you get a different manifestation. The same thing with PMF. And yet PMF and PV are different. So again, it's the context of the patient, not the specific mutation. Okay, and just to show you clearly here why these diseases should be separated, they each have different survival times. So let's go back to good old polycythemia vera, the most common of the myeloproliferative disorders. And this is a patient of mine who would be called in the old days the spent phase of the disease. So you can see the red cells. And then the next layer are his white cells. His white count was 400,000, hardly spent. And the next clear layer is actually all his platelets. And he had about 600,000 platelets. And the top is his plasma. But actually, that whole thing is his plasma. So you can see the plasma is expanded in this disease to accommodate what else is going on. So erythrocytosis is the distinguishing feature for this disease. But of course, the World Health Organization says we really don't have to worry about that. We have distinct criteria. And as I say, there's no specific marker. So some unresolved problems in polycythemia vera would be this. Is this a monolithic disease that I'll show you that everyone has the same clinical course? Or is this heterogeneous, that some people are going to have a much different clinical course than others? And this we know is true in primary myelofibrosis, so why shouldn't it be true in polycythemia vera? And we know there are differences between men and women with polycythemia vera. What's the story with that? And finally, what you came to hear, I think, is can we stratify these diseases to see who should get what therapy? So here we go. So Louis Wasserman was one of the early pioneers in the study of polycythemia vera, and he put together this natural history, which starts out with people are feeling well, then they have a headache because they have too many red cells or they get a blood clot, and then things get, well, they're not making as many cells anymore, so-called spent phase, which really doesn't exist, and then finally they get myelofibrosis, and then they get acute leukemia. Now, to Dr. Wasserman's credit, he said, I hypothesize. To the discredit of the hematology community, they took that as gospel, and it went from a hypothesis to the truth. Not his fault. So, that's the first point. Monolithic or heterogeneous. Now, I won't have time to go through all the things that women do differently in this disease, but they do so many different things that why people haven't signaled this out as a cardinal feature of the disease is beyond me. And I also get angered at women physicians because when they're in the audience, I always say, you know, get with it, and they don't. But let's leave that as it is. And finally, are there different kinds of polycythemia vera? Well, yes, there are. You take care of enough patients, you'll find some patients who manage to get their hematocrit up to a high value, and then you phlebotomize them, and you can keep phlebotomizing. And I inherited this patient in the 70s and lost her in the 90s. But she was well into her 80s, and we just come back to clinic. And then there are patients who come in, and they have a very big spleen or a big liver, and they are in trouble. And that happens pretty early. I think there are differences. So, how do you get at this? Well, you've had the nice genetic diagrams, and I just put a cartoon of a cell that could go back to high school biology with its plasma membrane, protecting it from its environment, keeping it as an intact unit, and the cytoplasm and the nucleus, and a receptor, and you've heard all this. And so either a ligand comes up to the receptor or the receptor is activated, and it sends transcription factors to the nucleus, and they sort of act like scribes or stenographers, and they will transcribe the DNA. Now, the DNA, as you've heard, well, there are about 22,000 genes in our DNA, which is only about 1% of the DNA, actually. And we can sequence it, and you heard Dr. Crispino talk about deep sequencing to get down and find out what are the mutations. And the good news is we can do that. The bad news is there's usually about 17 or 20 of them. So which one is important and which one is not? So that's a problem. But DNA is just like somebody sitting here and they happen to be an expert in something, and I don't know it unless I interrogate them and they talk to me. That's transcription. So when the DNA is transcribed, we get the message RNA. Now we can measure the amount of RNA in a cell. We can measure the sequence of RNA. Now we're getting into something interesting because a mutation has to be expressed, or if it's silent, it doesn't mean anything. It's the old story, if a tree falls in the forest and there's no one there, does it make a noise? So RNA is where the noise is made, and we can measure the number of transcripts coming from the DNA. So what we can measure is what genes are turned on or turned off, okay? And then we get to protein. Protein is much more complex, so we like to, and it's the actual proteins that tell the cell what to do, but that is sufficiently complex. But it's easy to get at the RNA to tell us what the DNA is doing. And this is called gene expression profiling, where we ask, in a malignant cell, what genes are turned on compared to a normal cell? And we can compare them. And we can distinguish malignant cells from normal cells, even though they look exactly the same. So in the subtype of a tumor, which looks all alike, their biologic behavior can differ. And we can use it for risk stratification to see who needs treatment and who doesn't. So we applied this to Polycythemia Vera. The idea of gene transcription was not original to us by a long shot, but it was a great technique. You can get plenty of stem cells, the most primitive stem cells, out of the peripheral blood in patients with Polycythemia Vera. That would be the subject of another talk. And we can get them from normal volunteers or people who sell their blood. And we can purify these stem cells so they're viable, and there's no other cells around to complicate affairs. And then we use the RNA from these stem cells, and we ask, what genes are turned on? Because when a gene's turned on, it makes an RNA transcript. So that's the experiment. And just for your viewing pleasure, here's what a normal stem cell and a PV stem cell look like. They look identical, and the CDs are what are called cluster designations. They have these particular two proteins that we use as markers of stem cells. Both of these stem cells have the same markers. They both are immunophenotypic. In other words, you couldn't tell the PV stem cell from a normal stem cell by just looking at it or doing the surface markers on it. So they're very much alike. Now, this is hard to see, but I just want to show you the data. We took a population of 19 patients. Now, how did we get these patients? Well, as they came in the door and they were willing to participate in the study and give us blood, we took their blood, and you need a certain number of stem cells to do this study. So the selection here is people who have the most stem cells, which is really serendipitously important for the reason that people who have fewer stem cells have a different disease than people who are clonally dominant. So every one of these patients was clonally dominant, and every one of these patients was identical with respect to age disease duration, Jack2 V617F allele burden, their hemoglobin levels, their white count levels, their spleen size. The platelets were different in the men because they tended to have more chemotherapy, though most chemotherapy was very mild. And what we did was we said, you know what, women are not small men with polycythemia vera. And we compared the gene expression in women patients with normal women. Seems like it makes sense. And men with normal men. And if you do that, something really interesting happens. Female patients with polycythemia vera dysregulate 251 genes. Now, there's 22,000 genes, and you can do the math. We're down about 1% or less than 1% of the genes. That's all it takes. The men, on the other hand, dysregulate twice as many genes, so about 2%. And that's all. So then you ask the question, well, women dysregulate fewer genes than men. Do any of them dysregulate the same genes? Do the men and women? And guess what? There are 102 genes that both men and women dysregulated. Either made too much of the RNA, transcripts where the gene was very active, or the gene was shut off. And as Ron and John told you, this is the epigenetic part, shutting off of genes. So we have two things going on here. So we took the 102 genes, and then we did an experiment that, and this is somewhat complex, but what you ask here is, can we mathematically, with these 102 genes, separate polycythemia vera patients into groups? We had no idea. And this is sort of, the best way I could explain this is rolling the dice. It's called unsupervised hierarchical clustering. And what you do is you go through and ask on individual genes, and the individual genes are down at the left side, their names are on the right side. Which of these genes is up or down regulated in a given patient? And when you do that, you end up with what you see as a tree. And any of us who've studied evolution, this is like an evolutionary tree. And what you find out with the arrows is, if you look up the top, the tree splits into two groups, okay? And the two groups are a little different because they are different branches of that same side of the tree, but nevertheless, they're on one side of the tree. And then there are two groups. I have the blue group, and on the other side is a red group and a green group. Well, the green group, I'll give you the answer, those are normals. And you'll notice that one of the polycythemia vera patients drops into normal. That's how close related in these stem cells normal is to polycythemia vera in that group of patients. And so each of those little lines up top represents one of the 19 patients and or the 6 controls. So who are these patients? Well, we got blue and red again, okay? Sort of like the politics, and I didn't do this intentionally, but it sort of worked out that way. So if you look, male and female are out of this now. Male, we've taken them, gender is out now. The ages are not important. The Jack2 allele burden, which everyone says is really important to tell you what's going to happen to you, not important. The leukocyte count, which everyone says we've got to control the leukocyte count, not important. In this series, the platelet count, even though these numbers look different, there's a spread, which I haven't shown you. It's not important. But what was important? Well, the red group was more anemic. And that's the group that was closest to normal. And disease duration was, you can see how wide the ranges are. They're pretty close to agreeable. But there were more thromboses in the red group. There was bigger spleens. There were more splenectomies. There was more chemotherapy. There was transformation to AML. And the transformation to AML, I'm going to say out of self-defense, was the only patient in the blue group that got chemotherapy. And surviving, you can see, and we took us, 6-7 years to do this study. Thank God for Department of Defense and the NIH, otherwise this wouldn't happen. So we have something very interesting here. So who are these groups? Well, one group of patients is a very benign disease. They live a long time and they don't have a lot of problems. The other group doesn't live as long and they have a lot of problems. So we then said, well, can we narrow down that 102 genes to a small number? So we got down by another mathematical manipulation that we could distinguish these two groups by 30 genes. So now we're getting something workable. And if you keep doing the math, you can get down, let's see if we can get down to 10 genes. And so we set up an assay, and the assay just says, if the 10 genes on the left side are upregulated more than the 10 genes on the right side, or there's actually 6, but it's the 10 genes, you get a score. And if you get 1 point if it's twofold increase, the gene expression, and if you have a score of 5/6, you have bad disease. So we then did a clinical score using the same phenotype that we saw in our patients: anemia, spleen size, splenectomy, major vessel thrombosis. We're not talking about ocular migraine or erythromelalgia, chemotherapy or immunomodulation, or acute leukemia. And then we went back and we took 30 other patients, unselected patients, pulled out of our patient cell bank by people, not me, totally blinded. No one knew who these patients were. They just had good RNA. And they're essentially similar in every respect. There's no differences between them statistically. And then we did the same 10, we took the 10 genes and said, can these genes split these patients into 2 groups? So it's all done blind. And the answer is it can. No difference between men and women, no difference in age. There was now, if you'll see, we've got someone out there who's now 40 years with PV that's, I think, skewing things. But the fact of the matter, disease duration was different. The allele burdens weren't different, okay? But if you look at the 10 gene screen scores, they were split perfectly, okay? And if you look at the clinical scores, the same thing happened. So we can, with 10 genes, say who has aggressive disease and who does not have aggressive disease. And then this is the same cohort. And if you look again, gender, age, leukocytes, Jack2 V617F burden, don't matter. Thrombosis, anemia, disease duration. Now, this time platelet count came out in favor of the aggressives. Palpable splenomegaly, spleen size, chemotherapy, clinical score. We got the same thing that we got when we did the 102 gene screen. So now we think we have an assay that can tell us who has aggressive disease and needs therapy and who just has indolent disease and should stay away from chemotherapy at all costs. And what this slide shows, it's hard to see. We've been able to follow some of these patients for as long as 6 years. And if you have an indolent score, you stay indolent, except for one patient so far. And this is the one patient who was not a survivor over the past 6 or 7 years in the indolent. And she had a score that was 2. And then 2.5 years later, we repeated it in her, and her score was 6. And we repeated it again, and it was 5. And otherwise, as Ron was talking about the patient with the leukemia cutis, everything else looked normal. What's going on here? Well, it turns out 6 months after, she looked perfectly normal. 6 months after that score, she developed acute leukemia. So we don't have more patients in terms of this sort of thing, because we're doing more right now to see where this is. But what we think is the following, that if you do gene expression and control for sex as a confounder, you can get a set of genes that is involved in Polycythemia Vera. And I will tell you, we have the myelofibrosis genes. And you have 2 types of myelofibrosis. You have the benign type in the indolent form, and then you have the aggressive type in the aggressive form, okay? So they all get myelofibrosis eventually, but they're different, okay? Now, the 102 genes, we can set people into 2 groups. Now we've got it down tenfold to a tenfold panel. And this keeps to continue correlating with severity score, so that we can think now that we can divide patients into those who need therapy and others who need no more than phlebotomy and occasionally aspirin. So polycythemia vera is not a monolithic illness. We have this informative panel. And the characteristics of this panel indicate it should be useful in identifying patients who would most benefit from therapy. And I thank you very much.